Good morning, guys. Sorry, I just sent out the invitation for the Zoom meeting a few minutes ago. Forgot to do it last night. So can you hear me, Bo? Hello, everybody? Yes, yes, okay. Sounds good. So we are on chapter 11. Chapter 11 and almost chapter 12, almost there. Now there's good morning. Yes, good morning to you, Christopher. And let's discuss our cell membranes. Let's discuss specifically how we're going to transport things across cell membranes. And that's the chapter, that's the subtopic of our discussion today and on Monday. Perhaps on Wednesday, we'll see how it goes. A very important topic. Signaling is, which is the last chapter. Obviously, signaling is a huge topic. So we will only look at some examples of signaling in a cell. So even though we're going to start on Wednesday, but we may not have to. Well, we don't have to. So cell signaling is just, we're going to just cover some examples. We're just going to look at some particular cell signaling pathways, but but specifically how the signal is delivered across cell membranes. I think that could be more useful for us. So, okay, so let's do it today, Monday, and perhaps Wednesday. We'll see how it goes. Okay, so do you have any questions from the lecture on Wednesday? From the lecture on Wednesday? Anybody? All right, so there are 22 of you right now. Okay. Well, keep in mind, you can ask questions at any time.
Wait, I have a quick question.
Yeah.
Should we be able to read an electron micrograph of a cell membrane?
Electron micrograph. Well, there was a slide. Yes. So, well, I mean, you shouldn't be able to read it, but I think it's pretty clear. No, you're talking about the slide that we had that we looked at on Wednesday.
Yes.
This one. Yeah. Yeah. I mean, so. Just you should be able to see, okay, so this is the nucleus, right? So there's mitochondria. It's all shown to you, right? So you don't really need to read anything here. In the plasmic reticulum, you can see these wavy lines, right? Secretory granule. So secretory granule, so you know that those are important for production of, for example, cellular waste, right? Cellular waste and excretion of that into the extracellular space. Yeah, so it's not really, there's not much here. All right, so composition. So we talked more about that, and we're going to be talking about this for quite a bit today and on Monday. Composition of cell membranes. So obviously the lipid composition. So we talked about phospholipids, right? So glycerophospholipids and sphingolipids as the primary ones. but not necessarily. There's sterols. We talked about cholesterol and sterol-derived molecules. So the composition changes depending on the organism, depending on tissue, depending on organelle. So we're gonna look at on the next slide. We're gonna look at the how this composition changes depending on the particular type of cell membrane. Now, abundance of sterols varies, right? So sterols, remember those molecules like cholesterol that pack the cell membrane and change the viscosity. So they may change the fluidity of cell membranes. So you already told me. I asked you, and you knew that cholesterol makes cell membranes more rigid, for example, right? So now sterols are, so many of them are not present in prokaryotes, right? In prokaryotes, and cholesterol, for example, not only it's absent in prokaryotes, it's also absent in mitochondria. So mitochondrial membrane. So cholesterol is also absent there as well. For example, galactolipids. So those are lipids which have galactose, right, as their head group. So we talked about those, and those are abundant in plant chloroplasts, but almost absent in animals. So here is the table. Some of what we talked about is put in numbers. So for example, this is a human myelin sheath. So remember, a myelin sheath is the specifically responsible for the insulation of the nerve signal transduction, right? And so you can see here there's not so much protein. Primarily, there's phospholipid and sterols, cholesterols, right? So myelin sheath is very fatty. It's very fatty, not too much protein. You can see the other cell membrane, the other membranes have much more protein compared with myelin sheath. Myelin sheath, the primary role of myelin sheath is the insulation. And so you can see here, there is not so much protein, primarily phospholipid and sterol. Lots of galactolipids, lots of plasmalogens. Now, plasmalogens, remember, those are that have ether, right? Ether lipids, lots of fatty stuff, lots of hydrocarbon. Now, this is mouse liver. So there's much more protein, so lots of functional stuff. So remember, proteins will be responsible for delivering the signal across the cell membrane, for example, right? And you can see here, there's much more protein in mouse liver compared with the human myelin sheath. Yeast, lots of protein. Yeast is a unicellular organism, needs to communicate with the outside world. Lots of protein and very little phospholipid. So only 7% of phospholipid and 4% of sterol. But as far as sterol, something that we need to discuss in more detail, maybe a little later. We'll talk about ergosterol. So in our cell membranes, we have cholesterol is the main ingredient. In yeast, it's ergosterol. Ergosterol and cholesterol they have similar structures, but not quite. So ergosterol also has this similar 4-ring-based nucleus, but it has more double bonds. So ergosterol has slightly different structure. And in fact, actually, many antifungal, so yeast is obviously, it's a type of fungus, right? So many antifungal drugs have a mode of action based on the inhibition of biosynthesis of ergosterol, right? So if the fungal cells do not have enough ergosterol to make the cell membranes, obviously they will not be viable. And so many, for example, amphotericin B. Amphotericin B is one of the main antifungal drugs that is used in the clinic right now. It actually binds to ergosterol. It binds to ergosterol, but not to cholesterol, and it shows specificity for fungal membranes rather than for eukaryotic membranes. All right. So here is membrane composition. So here, now I'm not so good with colors, but I can see this major color, the blue and dark blue, right? So this phosphatidylcholine as phosphatidylethynolamine. These are main ingredients. So these are main components of cell membranes across the species, across the different types of organelles. But as far as the precise composition, that varies. So you can see in the plasma membrane. So you can see there's lots of cholesterol. I think I can tell, even though it's not the same. I cannot tell the colors very well, but this is the first one and this is the first one, right? So cholesterol is one of the main components of cell membranes, but it's virtually absent in mitochondria. Virtually absent in mitochondria, but lots of it in cell membrane. Lots of it cell membrane. Cardiolipin, there is lots of it in mitochondria. So this is the characteristic of this lipid. Remember I told you about cardiolipin. So this one is a kind of a dimer, right? It has 4 fatty acid residues as opposed to 2 fatty acid residues. And lots of it is in mitochondrial cell membrane, but not too much. In fact, virtually none in the cell membrane. All right, any questions about this? Keep going. Now, what's important is also not just the composition, but also asymmetry. What this stands for is the presence of the lipid in the outer layer versus the inner layer, right? So if we have for example, plasma membrane, it's a bilayer, right? So there is also some lipids are present in the outer layer, and some lipids are more present in the inner layer. And so this is the distribution. Now I will not attempt to describe to you which ones are present, which ones are absent in the inner layer and the outer layer. But for example, this one, I can tell you, it's actually phosphatidylserine. Now this one is important because it's a signal type lipid. So normally cell membrane does not, the outer layer of cell membrane does not have phosphatidylserine, right? And so, but when a cell undergoes suicide, so for example, apoptosis, right? So we talked about apoptosis many times. And one of the characteristic features of programmed cell death, of apoptosis, is the appearance of phosphatidylserine in the outer layer of cell membrane. So, so the way apoptosis works is obviously a cell that undergoes programmed cell death is telling other cells in the body, such as phagocytes, is that here I am, you know, I'm killing myself. Please come here and eat me, right? So phagocytes, which are types of cells which come and clean up all sorts of mess, right, in the body, making sure that no cells are debris are present in the body, right? So the phagocytes come up and they basically eat all the debris, right? Making sure that there's nothing left. And so how do they know? How do they know which cells need to be eaten? Are those cells which have phosphatidylserine in their outer cell membrane. In their outer cell membrane, they know that these cells undergoing apoptosis, they need to be eaten. They need to be not left behind, and making sure that no mess is left behind, right? Okay, so this is one particular type of lipid, but there are obviously others, right? There are obviously others. You can see there's the banana-like type structures. These are cholesterol, right? Cholesterol, then there is OH on the lipids, so that's ceramide. Ceramide, when you have an OH, right? So you know the structure of ceramide. You know the structure of ceramide, there is an OH in the structure, and things like that. Okay, any questions about the composition and symmetry? So that's as far as the symmetry. So why we have asymmetric distribution of different lipids, right? So two leaflets. So leaflets referring to different layers of cell membrane. Yes, and also phosphatidylserine when it's on the outside, it also activates blood clotting. So depending on what type of cells, if it's on any cells, it marks cells for cell destruction, but it's also absent. It's also present on platelets, and when it's present on platelets, that means it participates in blood clotting, right? So platelets are activated. Archaea. Now, archaea have multiple constituents. So now keep in mind that glycerol has a chiral center, right? So you can have so two fatty acids and the phospholipid. So remember that one. So this is a chiral center, right? This is a chiral center. You can see it has oxygen, CH2, oxygen bound to two different types of substituents, right? So here, if you go to the left, it's bound to the ester of a fatty acid. If you go right this way, you go to the phosphodiester, right? So so this is a chiral center. This is a chiral center. And so phospholipids are chiral. So just like amino acids, we talked about chirality. Lipids are chiral in organisms. And you can see here, for example, in archaea, it's primarily L-glycerol, right? So it's and in bacteria, it's D-glycerol. So as far as fatty acids, in archaea, it's primarily branched. So in fact, we're gonna look at on the next slide. I'll show you how in archaea these fatty acids are branched, and in bacteria there are straight chain. Now unique linkages. So we talked about ether versus ester, right? So these are ester ones, and remember, if you don't have the hydrolyzable functional group, right? If it's not ester, if it's ether, then it will be an ether lipid. And also membrane topology. So so you can have a bilayer, right? Bilayer, where you have fatty acid and a head group, like so. Bilayer. But you can also have monolayer. For example, some archaea, they have a lipid which has two head groups. It has two head groups, has a long hydrocarbon residue and two head groups, like so. So this will be monolayer. Monolayer. Monolayer, and so this is present in some archaea. So for example, here's this type of structure. So diphenyl type of lipid. So you can see here, so this archaea, now it lives near volcanic hot springs, right? So why do you think, so those of you who like to think rather than memorize things, looking at the structure of this monolayer, and so obviously you've noticed that this is ether, not ester. Why do you think archaea, the type of organisms, why do you think they evolved using the ethers as opposed to esters for their cell membranes?
They're stronger at high temperatures?
Yeah, good job. So yeah, so this, what do you mean by stronger?
Like they're not hydrolyzed as easily?
Yeah, they don't hydrolyze easily, and so ethers have obviously stronger chemical bonds, right? So so phospholipids will have an ester here, and species that live at higher temperatures, which can be more hydrolyzed more easily, do not have esters. That's right. Yeah, so you see, it's all organic. OChem2. OChem2 can explain the entire biochemistry for you. The entire biochemistry. All right, and so you can see this one, now this is isoprenyl groups. So isoprenoid tetraethers Isoprene is from OChem 1, OChem 2. You also probably know that isoprene is 5-carbon type structure, which is responsible for production of high molecular weight polymers. So for example, you can polymerize this, right? You can polymerize this polyisoprene. Now, do you know polyisoprene? Do you know where it's present in nature? Polyisoprene. For example, in tree sap. I don't know if I'm spelling it correctly, but tree sap, right? So it's a natural polymer, polyisoprene, which can undergo polymerization. You can see here, you add some kind of radical. You add some kind of radical, and it will add to the double bond and undergo polymerization. And so this is a natural polymer and can give rise to lots of polyisoprene motifs. And you can see here, so some lipids, for example, in bacteria, which can actually live and obtain its nutrients, obtain its structural molecules from natural sources. You can see here, it's an ether-based lipid. All right, and you can see also here that it has sugars, right? It doesn't have phosphate, but has sugars. So this is a glucose and galactose, right? So glucose, beta 1 to 2, and galactose. So you know how to read this nomenclature. All right, so as far as proteins, so we have lots of proteins in membranes. So specifically, we talked about opsin or rhodopsin, right? So the function of opsin is obviously perception of light, right? Then you have hormones. So for example, molecules which respond to other small molecules on the outside, in the outside world. So for example, insulin receptor, right? Neurotransmitter they respond to acetylcholine. Pheromones respond to taste, smell. Different channels, gates and pumps respond to nutrients, ions. So for example, potassium ion. So we'll talk about ions on Monday, I think. So you can also have enzymes in cell membrane, right? So we'll in biochemistry 2, you will also encounter a lot of enzymatic action in the cell membrane. For example, ATP synthase. ATP synthase is an enzyme that is incorporated in cell membrane and whose function is to produce energy by synthesizing adenosine triphosphate. All right. So there are different types of membrane proteins, right? So there are integral membrane proteins. So you can see here. So these membrane proteins, now there are polytopic, those which span the entire membrane, right? So there is a small portion of it hanging out on one side and portion of it hanging out on the other side. So these are known as polytopic, right? Across the both bilayers. And there's monotopic. Monotopic are those ones that actually only embedded into one bilayer, into one layer of the cell membrane. And one way to get them out is obviously through the use of a detergent, right? So if you use a detergent such as a soap, you can see here, you can apply to it. You can apply to the cell membrane, and you can see here that the hydrocarbon tail of the detergent will form hydrophobic interactions with the integral membrane protein, this particular portion, this portion that normally inserts into cell membrane, right? And so this portion which inserts into cell membrane, which is in the middle of the protein, you can see here, that's where detergent will go, right? And not the polar portion, but the middle one. Now, there is also a type of proteins which are known as peripheral proteins. So you can see here, so these associate with the, for example, with lipids themselves, right? So for example, phosphatidylinositol phosphate. So it's a lipid. And you can see here through biological regulation, you can have a protein associating with the lipid. Now this one is, so it has an inositol and a phosphate. So obviously the protein that you're, that is of interest can interact with the negative charge if the inositol has phosphate groups, right? And you can see here, the protein of interest must have a negative charge, which will interact with the integral protein on the cell surface. And you can see here, so one way to isolate it is by changing the pH, for example, adding urea, breaking bonds, breaking hydrogen bonds, carbonate, right? So breaking the electrostatic bonds, chelating agent, and so on and so forth. All right, and GPI anchored protein, I keep promising you, but we will see actually GPI anchored protein slide in them today, maybe not today. If not today, then on Monday. So this is a glycosyl phosphatidylinositol anchored protein. So protein that is covalently attached to the membrane. All right, so so for peripheral membrane proteins, so these associate with the polar head groups of membranes, right? Relatively loosely. And you can remove those easily by disrupting ionic interactions, right? And you can disrupt those ionic interactions with high salt or change in pH. You can, so this one, for example, is amphitropic protein, right? So, so this one is linked to the cell membrane using this covalent attachment. And you can see here, it's attached to a lipid. It's attached to a lipid, right? So you can see there's a head group. There's a head group. There's a double tail of a fatty acid. Double tail of a fatty acid, some sugar, and the protein is going to be attached through an oligosaccharide. And there's a phosphate there as well. The phosphate will actually be hydrolyzed by an enzyme known as phospholipase C. Phospholipase C. So it's a GPI anchored protein. And so here is the structure. Here's the structure. So if this is the protein, right? So there are lots of amino acids here. And what do we have here is the carboxyl terminus. So this is carboxyl terminus of a protein, and we have ethanolamine, right? So, so this is ethanol. This is the amine portion of it, attached through a phosphodiester bond through a mannose. So there are 4 mannose residues. There's a N-acetylglucosamine. There is inositol. Inositol, phosphodiester bond attached to glycerol. Glycerol has 2 fatty acids embedded in the cell membrane. So, so this will be a GPI anchor, glycosyl phosphatidyl inositol anchor. So it's a very common biological mechanism for attaching proteins to cell membranes. And the enzyme that cleaves that is the phospholipase C, known as PLC. PLC, the enzyme that can cleave that. All right, so other types of ways proteins can be attached to cell membrane, for example, palmitoyl group can be attached through a cysteine. So you can see here, so cysteine forms an ester with a carboxylic acid, with a fatty acid, right? The fatty acid will be embedded in a monolayer, in a sort of single layer of a bilayer, and attached obviously through the cell membrane, attached to cell membrane using this kind of hydrophobic interaction. Now there's this myristoyl on amino terminal glycine. So you can see here, the cysteine was inside the protein somewhere. Glycine was on the terminus. Glycine was on the terminus, and glycine was had a myristoyl fatty acid attached to it, and can be linked to the cell membrane using that kind of interaction. And this one is farnesyl. Farnesyl is, you can see farnesyl has a, it's also an isoprenoid type of hydrocarbon tail, right? And so isoprenoids, the interesting thing is that actually many proteins utilize this mechanism for attaching to cell membranes, right? And so there are a lot of proteins which are responsible, for example, for cell division, right? And so there are a lot of cancer drugs. A lot of cancer drugs which have been developed that inhibit the biosynthesis of the farnesyl hydrocarbon tail, right? So a lot of them have been tested in clinical trials. In clinical trials, specifically to inhibit this kind of farnesyl attachment of proteins to cell membrane. And unfortunately, so far there has been no cancer drug that has been approved for human use that is based on this kind of mechanism. So it turns out that this kind of isoprenoid or farnesyl attachment of proteins to cell membrane is not only utilized by cancer cells, it's also utilized by normal cells. And so all these drugs are just too toxic. They're too toxic, and they don't differentiate between normal cells and cancer cells, unfortunately. It's all that's whenever you develop a cancer drug, that's the name of the game, right? Is you have to find something that some kind of mechanism that will make your drug selective, make your drug selective, and will kill or will stop the growth of cancer cells, but will not affect the normal cells. You know, unfortunately, these kind of drugs that inhibit biosynthesis of farnesyl tails, they are not selective. They're not selective, even though there was a lot of activity in pharmaceutical industry. Not recently, because now everybody is kind of already disappointed in these kind of drugs. But at the beginning of 2000s, I remember there was a lot of interest and a lot of enthusiasm in pharmaceutical industry on developing these kind of drugs, but never materialized. All right, so lipid anchors. So what do we have? Okay, so this is what we talked about: long chain fatty acids, isoprenoids, sterols, glycosylated phosphatidylinositol. Now this is not PGI, this is GPI. So I found on the outers, yeah, and the GPI anchors, one thing for us to remember, they're found only on the outer face of cell membrane. All right, farnesylation. So we talked about that, farnesylation. And for farnesylation, it's important that the protein that is attached to the farnesyl hydrocarbon tail, it has this kind of sequence, right? So C has to be cysteine, then 2 A's can be some kind of aliphatic amino acids, right? 2 of them, and X is usually one of these 4. One of these 4. And so the reaction that so the drugs which I was telling you about, many of them were developed against this particular enzyme, farnesyl transferase. Farnesyl transferase is the enzyme that will catalyze the attachment of the farnesyl tail to the cysteine residue on the protein. And so if you don't, so the proteins which do not have this farnesyl chain, right, they do not go to the membrane. They do not go to the membrane, and they will be inactive. And so RAS is one type of protein. So that is utilized for signal transduction. When we talk about signal transduction, we'll talk about RAS protein. And so when we see onco, that means promotes cancer, right? So onco-RAS protein, it used to be a promising cancer therapy, but right now people are not so enthusiastic about this onco-RAS anymore. All right, so integral membrane proteins. So these ones, we had a picture of that, right? So you can remove these through the action of a detergent. Through the action of a detergent, they can be polytopic, monotopic, and obviously they have a symmetry. So whatever you have on this side is different from what you have on that side, right? That makes sense. Makes sense. So integral membrane proteins, in most cases, these are alpha helical structures. Alpha helical, they have specifically, they have hydrophobic amino acids in this alpha helix, which will obviously interact with the fatty acids of the lipid bilayer, right? Lipid bilayer. And you can see here that, so for example, this is potassium channel. You can see here on the outside, so these are going to be charged residues, the blue ones, right? And the portion of the protein of the channel that spans the membrane is going to be hydrophobic. So this kind of silver type structure. And you can see here that lots of these proteins which span the cell membrane they similarly have the hydrophobic amino acids in the region that spans the cell membrane. And not all of these are alpha helical. Some of these contain beta sheets. Some of these contain beta sheets. You can see those, right? And you can see here. So what do we have? Phenylalanine, glycine, valine. All of these are hydrophobic ones, right? All of these are hydrophobic. And you can see now these ones, arginine, lysine. Now these ones are already hydrophilic ones, which now once the protein is on the outside, then you can have hydrophilic amino acids. But the part of the protein that is inside, here we have hydrophobic amino acids. And so this is oftentimes, you study this by what's known as hydropathy plots. So specifically, there is a measure for hydrophobicity known as hydropathy index, right? So for example, this protein, glycophorin, which spans the membrane only once, you can see there's a very nice, as far as the residue number. So this is a residue number, and this is hydropathy, right? So basically, whichever is in the positive region is hydrophobic, negative region is hydrophilic. So you can see here, so this protein only has one region, specifically spanning the residues between 70 and 95, right? So these residues mostly hydrophobic, and they will actually be located in cell membrane. And this one, Bacteriorhodopsin, this spans cell membrane 7 times. So in other words, if you have a cell membrane like this, right, it goes 1, 2, 3, 4, 5, 6, 7. So Bacteriorhodopsin crosses the cell membrane 7 times. And you can see here, there are 7. And so every time it crosses it, so for example, this region here, hold on a second. This region here, right, is the same as this region here. This region here is the same as this region here, right? So this is the region which will be exposed to the aqueous medium. And once it goes into the membrane, it becomes hydrophobic. Actually, this one and this one, I may be wrong. It's actually this one on the outside. Yeah, so if this is, let's say this is A, this is A. If this is B, then this is going to be B, right? And then this one will be C, and this is a tiny one, C there. All right, I think we should be done for today. Do you guys have any questions?
Could you go back one slide? Just like one slide where you like did the ABC. I just missed it on the graph.
You missed it on the graph. Hold on a second. This one?
Yes, thank you.
Okay. All right, guys, you have any questions?
I had a quick question. When we talked about alpha helixes last time, when we were first introduced to them, we talked about on the outside we had hydrophilic and on the inside we had hydrophobic. But when we're talking about the alpha helix and the transmembrane, they're only hydrophobic, outside and inside the helix.
Yeah, so it's a good question. So so for the alpha helix, so when we talked about different amino acids being parts of the alpha helix, we talked about amino acids interacting with each other, right? And not so much with the aqueous medium. So so in this case, they will all be hydrophobic because they are interacting with the lipophilic fatty acids, right? So right, so in this case, both it will be amino acids interacting with each other, and with the lipid bilayer of the membrane of the cell membrane. So all of these amino acids must be hydrophobic, yes.
Okay, because I just thought the like hydrophobic effect kind of like promoted alpha helix like formation and the interaction right between 1 and 4. So I just didn't know, like, how are they interacting if they're all like non-polar hydrophobic?
How they interact? Well, they're forced from the aqueous medium into the membrane bilayer, right?
Oh, okay,
because they're hydrophobic, they do not localize in the aqueous medium. They're forced from the aqueous medium into membrane bilayer. Yeah.
Okay, thank you.
I have a quick question.
Yeah.
So you mentioned that membrane proteins contain beta sheets. Is there any type of structural advantage having an alpha helix formation or a beta sheet?
No, I think well, both alpha helix and beta sheets. I think most commonly you find alpha helices, but sometimes you do find beta sheets.
Okay, thank you.
But mostly alpha helices. All right, guys. Have a good weekend. Let me know if you have any questions. Place some more thoughts into the discussion part of the Canvas, and I'll talk to you on Monday.